Hello friends, welcome to Medical Day. In first part we have seen the chapter Living World. In this video we will see the second chapter of 11th Ncert, that is Biological Classification. So without further delay, let us discuss the chapter Biological Classification. Since the dawn of civilization there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between eukaryotic and prokaryotic, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphological, a need was also felt for including other characteristics like the cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification systems for the living organism have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what group organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Let us see the characteristics of the five kingdoms. Characters. Five kingdoms. Five kingdoms are Monera, Protista, Fungi, Plantae, Animalia. Let us see the cell type. In Monera, Prokaryotes. Protista include eukaryote. Fungi, Protista, Fungi, and Plantae and Animalia. These four kingdoms include eukaryotic cell type organisms, while only Monera include prokaryotic. Cell wall. In Monera, cell wall is non-cellulosic, include polysaccharide plus amino acid. In Protista, present in some. In fungi, present with chitin. Plantae, present with cellulose. Animalia, cell wall is absent in Animalia kingdom. Let us see the nuclear membrane. Nuclear membrane present in all four kingdoms except Monera. Body organization. Monera and Protista both have cellular level of body organization, while in fungi it is multicellular loose tissue, in Plantae tissue, organ, in Animalia tissue, organ and organ system. Let us see the mode of nutrition. In Monera, autotrophic, chemosynthetic and photosynthetic and heterotopic, saprophytic, parasitic. In Protista, autotrophic, photosynthetic and heterotopic. Heterotopic, saprophytic, parasitic. Autotrophic, photosynthetic. Heterotopic, holozoic, saprophytic, etc. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Phylogenetic relationship means evolutionary relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the three-domain, and thereby a six-kingdom classification. You will learn about this system in detail at higher level. Let us look at this 5-kingdom classification to understand the issue and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, worm, gymnosperm, and the angiosperm under plantae. The characteristic that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together group which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotopic group, fungi, and the autotrophic green plant, though they also showed a characteristic difference in their cell wall composition. The fungal had chitin in their cell wall, while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plant and both having cell walls, with Paramecium and Amoeba, which were placed in animal kingdom, which lacked cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future to depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but in also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4. Let us see the first kingdom of 5-kingdom system proposed by R.H. Whittaker, that is kingdom Monera. Bacteria are the sole member of kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Handful of bacteria are present in handful of soil. They also live in extreme habitats such as hot spring, desert, snow, deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, cocci, the rod-shaped bacillus, bacilli, the comma-shaped vibrio, and the spiral spirulium. Here they have given the picture of the 4 types of bacteria: cocci, spherical, bacillus, spirulium, spiral, vibrio, comma-shaped. Though the bacterial structure is very simple, they are very complex in behavior. Compared to the many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotopic, that is, they depend on other organisms or on dead organic matter for food. Let us see the types of Monera kingdom, the Alky Bacteria and Eubacteria. First is Alky Bacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, halophytes, hot springs, thermoacidophilus, and marshy places, methanogen. Alky bacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals, such as cow and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Let us see the Eubacteria. There are thousands of different eubacteria or true bacteria. They are categorized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plant and photosynthetic autotroph. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cell called a heterocyst. Example: nostoc and anemia. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotopic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Here they have given a picture of a filamentous blue-green algae nostoc. Specialized structure present in nostoc is called as heterocyst for nitrogen fixation, and a sheath is mucilaginous sheath. Some are pathogenic, causing damage to human beings, crop, farm, animals, and pets. Chlorella, typhoid, tetanus, citrus, canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Some, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adapting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Let us see a dividing bacterium. At the time of dividing, they divide their DNA. Let us see the kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biologist may be a plant to another. In this book, we include chrysophyte, dinoflagellate, euglenoids, slime mold, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotic, the protistian cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Let us see the chrysophytes. This group includes diatoms and golden algae called as desmids. This is the important line. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water current. That's why they are called as planktons. Most of them are photosynthetic. In diatom, the cell wall forms two thin overlapping shells, which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatom have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years is referred to as Diatomaceous Earth. Being gritty, this soil is used in polishing, filtration of oil syrup. Diatoms are the chief producer in the ocean. Dinoflagellate. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cells. The cell wall has stiff cellulosic plate on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellate called a goniaulex undergo such a rapid multiplication that they make the sea appears red, called red tides. Toxins released by such large number may even kill other marine animals such as fishes. Let us see the euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and a long. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotopic by predating on other small organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Let us see the picture of euglenoids. First dinoflagellate and second euglenoid. Let us see the slime mold. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called a plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiate and form fruiting bodies, bearing spores at their tip. The spores possess true walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air current. Protozoans. All protozoans are heterotrophs and live as a predator or parasite. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, that is false feet. As in amoeba, marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness. Example, tryposoma. The sleeping sickness disease is called as trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that open to the outside of the cell surface. The coordinate movement of rows of cilia cause the water laden with food to be stirred into the gullet. Example, Paramecium. Sporozoans. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has staggering effect on human population. Let us see the kingdom fungi. The fungi consist a unique kingdom of heterotopic organisms. They show great diversity in morphology and habitat. You must have seen fungi on moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Tucinia is an important example. Some are the source of antibiotics. Example, penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animal and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent the food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular fungi, are filamentous. Their bodies consist of long, slender, thread-like structure called a hyphae. The network of hyphae is called as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called sinusotic hyphae. Others have septate or a cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotopic and absorb soluble organic matter from dead substrate, and hence are called saprophytic. Those that depend on living plants and animals are called parasites. They can also live as symbiont. in association with algae as lichens and with the root of fire plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia or sporangiospore or zoospore, and sexual reproduction is by oospore, ascospore, and basidiospore. The various spores are produced in distinct structure called a fruiting body. The sexual cycle involved the following three steps. First step is fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second stage is fusion of two nuclei called karyogamy. Third stage is meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cell. However, in other fungi, Ascomycetes and Basidiomycitis is an intervening dikaryotic stage. N plus N, that is 2 nuclei per cell occur. This is an important statement. Ascomycetes and Basidiomycetes possess dikaryon phase. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occur, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. Let us see the Phycomycetes. Members of Phycomycetes are found in aquatic habitat and or decaying wood in moist and damp places, or as oblique parasite on plants. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospore, motile, or by aplanospore, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by the fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamy or oogamous. Some common examples are mucor, rhizopus, that is called as the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. Let us see the Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example Penicillium, or rarely unicellular, example yeast, Saccharomyces. They are saprophytic decomposer, parasitic or coprophilous, growing on dung. Mycelium is branched and separate. The sexual spores are conidia produced exogenously on the special mycelium called conidiophore. Conidia on germination produce mycelium. Sexual spores are called ascospore, which are produced endogenously in sac-like structure asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarp. Some examples are aspergillus, clavicle, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and considered delicious. Let us see the Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracket fungi, or puffball. They grow in soil or logs and tree stump and in living plant bodies as parasites. Example rust and smooth. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus, mushroom, Ustilagus, smooth, and Puccinia, rust fungus. Let us see the Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phase of this fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkage were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of this member of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycites. The Deuteromycetes reproduce only by a sexual spore known as conidium. The mycelium is septate and branched. Some members are saprophytic or parasitic, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colotrichum, and Trichoderma. In kingdom fungi, you have to remember the septum, that is, the subdivision is septate or non-septate. Let us see the picture of fungi. First picture is of mucor, second is of aspergillus, and third is agaricus. The mode of spore formation you have to remember, and the spore is motile or non-motile. These are important points regarding the kingdom fungi. Let us see the kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll containing organisms commonly called a plant. A few members are partially heterotopic such as insectivorous plants or parasite. Bladder rot and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will be studying the eukaryotic cell structure in detail in chapter 8. Plantae include algae, Pryo, pterido, gymno, and angio. Life cycle of plant has two distinct phases, the diploid saprophytic and the haploid gametophytic, that alter with each other. The length of the haploid and diploid phases have and whether these phases are free living or depend on other vary among different groups in plants. The phenomena is called the alternation of generation. You will study further detail in kingdom of chapter 3. Let us see the kingdom Animalia. This kingdom is characterized by heterotopic eukaryotic organisms that are multicellular and their cell lack cell walls. They directly or indirectly depend on plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Slight features of various phyla are described in chapter 4. Let us see the various viroids, prions, and lichen, the last bit of this chapter. In the 5 kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroid, and prions. These are briefly introduced here. All of us have suffered the ill effect of common cold or flu, known what effect viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? That is the question. Viruses mean venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filtrate. M.W. Pichering in 1898 demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogen virus and called the fluid as contagium vivum fluid, infectious living fluid. M.W. Stanley in 1935 showed that viruses could be crystalline and crystal consists large of protein. They are inert outside their specific host cell. Viruses are oblique parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No viruses contain both RNA and DNA. A virus is a nucleoprotein, and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein coat called a capsid, made up of small subunit called a capsomere, protect the nucleic acid. These capsomeres are arranged in helical or polyhedral form. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptom can be mosaic formation, leaf rolling, curling, yellowing, vein clearing, dwarfing, and stunted growth. Here they have given the picture of bacteriophage and tobacco mosaic virus, that TMW. Here they given the RNA and the small subunits called capsid, and these are the capsomeres. In bacteriophage they mentioned here the head, collar, sheath, and tail fiber. Let us see the viroid. In 1971, Theo Donnier discovered a few infectious agents that was smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was a low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was smaller in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, that is BSE, commonly called as mad cow disease in cattle, or its analogous variant Creutzfeldt-Jakob disease CJD in humans. Lichens are symbiotic association, that is usually association between algae and fungi. The algal component is known as phycobient and the fungal component is known as mycobient, which are autotrophic and heterotopic, respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Let us see the summary of the chapter Biological Classification. In this, biological classification of plants and animals was first proposed by Aristotle. On the basis of simple morphological character, Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. In the five-kingdom classification, bacteria are included in Monera. Bacteria are cosmopolitan in distribution. These organisms have the most extensive metabolic diversity. Bacteria may be autotrophic or heterotopic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophyte, dinoflagellate, Euglenoids, slime mold, and protozoan. Protista have defined nucleus and other membrane-bound organisms. They reproduce both sexually and asexually. Members of kingdom Fungi show great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycitis, and Deuteromycetes are the four classes under this kingdom. The Plantae include all eukaryotic chlorophyll-containing organisms. Algae, Bryo, Pterido, Gymnosperms, and Angiosperms are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and sporophytic generation. The heterotopic eukaryotic multicellular organism lacking a cell wall are included in kingdom Animalia. The mode of nutrition of this organism is holozoic. They reproduce mostly by a sexual mode. Some acellular organisms like viruses and viroids, as well as the lichens, are not included in the five kingdom system of classification. In this way, we completed the reading of chapter Biological Classification. If you liked the video, then don't forget to subscribe to my channel called Medical Duo. Thank you.